it's important to point out that a new form of pyruvate kinase, namely the M2 isomer, has been discovered in recent years, and it's been found to be a moonlighting protein kinase in cancer. This is a totally unexpected observation by biochemists, and it turns out that this M2 isoform is the dominant form of pyruvate kinase in proliferating cells like tumors. New cells always express M2 first and switch to other forms during tissue differentiation. However, a shift back to the M2 form occurs during tumor formation itself. The pyruvate kinase M2, normally tetrameric, can dissociate to dimers that localize in the nucleus. And these dimers have low pyruvate kinase activity, but remarkably, they act as a protein kinase, and they use PEP as the phosphorylation substrate. Now, this is just incredible, and it was a complete surprise to the biochemists because there were very few cases of anything but ATP being used as a phosphorylation substrate in a protein kinase reaction, but this particular pyruvate kinase uses PEP as the phosphorylation substrate. The pyruvate kinase M2 protein kinase activity is essential to cell proliferation, and there are at least 100 proteins that are phosphorylated by this very interesting PEP-dependent protein kinase. Youngsam Lee and colleagues have shown that the pyruvate kinase M2 protein kinase activity is stimulated by succinyl 5-aminoimidazole 4-carboxamide 1-ribose 5-phosphate, or SACAR, and we will see in Chapter 26 that SACAR is an intermediate from purine biosynthetic pathways, so this is really a way to tie the synthesis of purine nucleotides to glycolysis, another remarkable observation of the interactions of pathways. Protein kinase M2, which acts to direct glucose to the pentose phosphate pathway for ribose 5-phosphate synthesis, also acts through its protein kinase function to activate expression of the genes for purine biosynthesis when nucleotides for DNA and RNA synthesis are needed, and that's really a hallmark of proliferating tissues such as tumors. There's the structure of SACAR. We'll see this again in Chapter 26, and we'll talk about this interesting phenomenon of the interaction between the purine biosynthetic pathway and glycolysis itself. Section 18.5 of your text talks about the metabolic fates of NADH and pyruvate that are produced in glycolysis. NADH is produced in reaction 6, and it can be recycled via either aerobic or anaerobic pathways. That NADH represents energy. The reduced coenzyme is energetic, and it provides electrons for a number of processes in the cell. It has two possible fates. If oxygen is available, that is under aerobic conditions, NADH is oxidized in the electron transport pathway, making ATP an oxidative phosphorylation. We'll see this in great detail in Chapter 20. In anaerobic conditions, NADH is oxidized by lactate dehydrogenase or alcohol dehydrogenase, providing additional NAD+ for more glycolysis. This slide shows the recycling of NAD+ and NADH. At the top, you see the glyceraldehyde 3-phosphate dehydrogenase reaction. At the bottom, the alcohol dehydrogenase reaction. And in this combination of reactions, glyceraldehyde 3-phosphate is turned into 1,3-bisphosphoglycerate, proceeds on in glycolysis to make pyruvate, and then the pyruvate can be decarboxylated to make acetaldehyde, which goes through the alcohol dehydrogenase reaction, which is essentially a reduction, and the NADH is oxidized and produces NAD+, which can go back and stimulate more of reaction 6. The picture on the right shows a mash of corn and other grains being fermented by yeast, producing ethanol and CO2, which can be seen bubbling to the surface. This is actually a vat of a mash that's going to be turned into bourbon, and this picture was taken at the Woodford Reserve Distillery in Lexington, Kentucky. Now, this same recycling of NADH is also involved both in oxygen-depleted mussels and also in hibernating turtles of all things. And so you see the same sort of pathway as we saw on the previous slide. The only difference is, at the bottom, we use the lactate dehydrogenase reaction to turn pyruvate from glycolysis into lactate. In this previous slide, we showed that pyruvate is first converted to acetaldehyde, which can go through the alcohol dehydrogenase to produce ethanol. In oxygen-depleted muscle, NAD plus is regenerated in the lactate dehydrogenase reaction. And it's also interesting to note that hibernating turtles can become anoxic and convert glucose mainly to lactate. Their shells release minerals to buffer the lactate throughout the period of hibernation. And this is very important because if they didn't have the buffering effect of the minerals in their shell, the lactate buildup over the winter while turtles are hibernating would kill the turtle. So this is a way that the turtle actually uses its shell during the winter period in its hibernation to protect itself. We always think of turtles using the shell to protect themselves from attack by other creatures, but in fact the turtle also uses the shell during the winter to protect itself from lactate. So what about the pyruvate produced in glycolysis? It also represents energy, and it has two possible fates, either aerobic or anaerobic paths. In aerobic conditions, pyruvate proceeds through the tricarboxylic acid cycle, the TCA cycle, which we'll see in Chapter 19. Anaerobic metabolism of pyruvate leads to lactate in microorganisms and animals, or ethanol in yeast, as we've just seen. And these are examples of fermentation. And here's the official chemical definition of fermentation: the production of ATP energy by reaction pathways in which organic molecules function as donors and acceptors of electrons. That's a definition of fermentation that you can use down the road. As I mentioned in your book about the Warburg effect, or Warburg effect, and cancer, this goes back to the early work of Otto Warburg in 1924, where he found that rapidly proliferating cancer cells metabolize glucose mainly to lactate, even when oxygen is plentiful. More recently, Lewis Cantley at Harvard has suggested that this behavior arises because cells need more than ATP. They must synthesize large amounts of nucleotides, amino acids, and lipids, and this requires lots of NADPH for biosynthesis, as well as intermediates for building blocks. Cancer cells divert large amounts of glucose to the pentose phosphate pathway, as we'll see in Chapter 22, to produce NADPH. And we'll talk about this when we get to Chapter 22. Here's a picture of Otto Warburg in his laboratory. He won the Nobel Prize in Physiology or Medicine in 1931 for his discovery of the nature and mode of action of the respiratory enzyme cytochrome oxidase, a bit of his work that really had nothing to do with glycolysis directly. And we'll see cytochrome oxidase in Chapter 20. Here's a look at the Warburg effect and cancer. Here's the pathways involved. We'll come back to these reactions when we talk about the Warburg effect in Chapter 22. So we're going to put this off for a little bit, simply say that signaling proteins and pathways regulate the glycolytic pathway. You can see glucose at the top of the diagram. Here's pyruvate at the bottom. So this is the glycolytic pathway, top to bottom. And you're seeing some of the steps that regulate glycolysis. Cancer cells route up to 90% of their acquired glucose and glutamine into lactate and alanine, producing large amounts of NADPH. One of the most important parts of this chapter is the understanding of how cells regulate glycolysis. This really is elegant evidence for regulation, and the following figures will show you this. Look at the standard state delta G0 prime values and notice that they provide little insight really into the actual free energy changes that occur in glycolysis. On the other hand, when we look at the plot of cellular delta G values, we learn something really, really interesting. Most of the values of delta G in the cell for the 10 reactions of glycolysis are near zero, but 3 of the 10 reactions have large negative delta Gs. These 3 reactions with large negative delta Gs are sites of regulation. They are hexokinase, phosphofructokinase, and pyruvate kinase. And regulation of these 3 reactions turns glycolysis on and off. Let's take a look at the graphs. First of all, here's the delta Gs at standard state, the delta G0 primes. And as you look across the graph, steps of glycolysis 1 to 10 and also the lactate dehydrogenase reaction at the end, the delta G0s are all over the map, so to speak. Some are negative, some are very positive. There's no real consistency or clear point in all of this. But if you look at the delta Gs in the cell, the cellular delta Gs, you see something very, very interesting. And once again, we'll ignore the lactate dehydrogenase reaction at the end here. But notice that except for reactions 1, 3, and 10, all of the delta Gs are very close to zero. In fact, they're essentially zero. the measurements in cells are difficult to do, and we can imagine that even though there are small delta G's, positive or negative here, that they're probably really effectively zero in the cell. And reactions 1 and 3 and 10, on the other hand, are large negative delta G's. Now, we've said before that the points of regulation in glycolysis are reaction 1, the hexokinase reaction, reaction 3, the phosphofructokinase reaction, and reaction 10, the pyruvate kinase reaction. We pointed out that the hexokinase reaction is not the main point of regulation for glycolysis, and that really is reaction 3, the phosphofructokinase reaction, and it's worth asking why that is. We mentioned it briefly before, but we should emphasize it here. PFK is the committed step in glycolysis. When this reaction occurs, all the other reactions go forward to make pyruvate and make ATP. So we call that the committed step. It's the step that says there's no other way to go after that. So this is the primary point of regulation in the pathway. And then the pyruvate kinase reaction is regulatory because we pointed out pyruvate is a branch point. But the point of this slide is that if you could simply turn reactions 1, 3, and 10 on or off together, you would turn glycolysis itself on or off. These three reactions drive glycolysis forward with large negative delta G's. If these reactions could be inhibited, glycolysis would turn off in the cell. And that's a very important point of understanding about the regulation of glycolysis. There's a very interesting box in Chapter 18 about tumor diagnosis using positron emission tomography. And if you're interested in a career in medicine, it's really important to understand this. As you know, Warburg showed early in the 20th century that tumors show very high rates of glycolysis. And this observation is really the basis for tumor detection by what's known as positron emission tomography, or PET scanning. Now, this involves the creation of antimatter particles, and we'll take a look at that in just a minute. Metabolites labeled with fluorine 18 can be taken up by human cells in the brain, for example, and then decay of fluorine 18 results in positron emission, the creation of antimatter particles. Positron-electron collisions produce gamma rays, and detection of those gamma rays with gamma ray cameras provides 3D models of tumor extent and location. So the substrate that's normally used for this, 2F18-fluoro-2-deoxyglucose, is really a substrate for hexokinase and shows where the high rates of glycolysis are located in the brain. This slide shows what happens when fluorine 18 is used as a probe in a study of tumors in the brain, for example. Notice that when fluorine 18 decays, the emission of a positron from fluorine 18 turns it into an oxygen. A proton in the fluorine 18 has given up a positron. It ends up with essentially the same mass because the mass of a positron is so small, and that turns a proton in fluorine 18 into a neutron, which makes the element into oxygen 18. Whenever positrons are emitted, they will collide with the first electron that they come to, and that creates the gamma rays that you see here that are emitted approximately perpendicular from the collision itself. Positron-electron collisions produce gamma rays. Nearly every major hospital now has a PET scanner, so every time you go past a hospital, just think for a moment that antimatter is being created in that hospital in order to scan and image the brains of tumor patients. Here you see the result of a PET scan on a patient with a tumor in the brain. The tumor is the red portion on the lower left of the brain, and that area is where large amounts of glycolysis are occurring. That's being detected by the concentration of fluorine 18 glucose in the brain itself. Section 18.7 in your text asks, are substrates other than glucose used in glycolysis? And the answer turns out, of course, to be yes. Sugars other than glucose can be glycolytic substrates. Dietary fructose, in fact, is degraded almost exclusively in the liver in an unregulated process that circumvents the PFK reaction. Mannose is routed into glycolysis by fairly conventional means, and galactose is more interesting. It's part of what's called the Lewar pathway that converts galactose to glucose, sort of, as we'll see. Let's take a look at each of these. Fructose metabolism in the liver is unregulated. There's a special aldolase in liver, a class 1 aldolase called aldolase B, which is unique to the liver, and it converts fructose 1-phosphate to glyceraldehyde, which must be phosphorylated by triose kinase to continue through the glycolytic pathway. Mannose and galactose can enter the glycolytic pathway via hexokinase ahead of the PFK reaction, so their catabolism is subject to the same regulation as glucose. Galactose metabolism is shown here. Galactose is first converted to galactose 1-phosphate by the galactokinase reaction, and then it undergoes a bit of a cycle where the galactose 1-phosphate uridylyl transferase converts the galactose 1-phosphate to glucose 1-phosphate, sort of. What it really does is it transfers the galactose 1-phosphate to a UDP molecule, and it exchanges glucose and galactose on that UDP. So the uridine diphosphate drops a glucose and picks up a galactose, which is then recycled by the UDP galactose 4 epimerase from galactose to glucose, and then that glucose goes on in the cycle of the galactose 1-phosphate uridylyl transferase to glucose 1-phosphate again, and on into the phosphoglucomutase activity going forward to make glucose 6-phosphate. It's very important to point out here that the two molecules shown to the right of this diagram, UDP glucose and UDP galactose, are what are referred to as sugar nucleotides, and they were first discovered by Louise Loire working under very difficult conditions in Argentina. Here you see the synthesis of UDP galactose from UDP glucose and galactose 1-phosphate. It's essentially an exchange of the glucose for a galactose. It involves what's known as a ping-pong kinetic mechanism. We didn't discuss those in Chapter 13, but they're described there if you'd like to go back and review it. This slide shows you how galactose 1-phosphate and UTP can combine to produce UDP galactose. This is the synthesis of a sugar nucleotide. And by the way, the synthesis of this sugar nucleotide releases pyrophosphate. The pyrophosphate itself is a high-energy molecule. It is a phosphoric anhydride, and it represents energy. And pyrophosphate breaks down on its own without the involvement of an enzyme in most cells and releases energy. The phosphoric anhydride linkage is cleaved to produce 2 phosphates, and this provides energy that can drive related and coupled reactions. Glycerol can also enter glycolysis, and this shows you that glycerol plus ATP can form glycerol 3-phosphate. Glycerol is produced in the decomposition of triacylglycerols, and it can be converted to glycerol 3-phosphate by the enzyme known as glycerol kinase. Glycerol 3-phosphate is then oxidized to dihydroxyacetone phosphate by the action of glycerol phosphate dehydrogenase, which is shown here. Glycerol produced in the decomposition of triacylglycerols then is a substrate for glycolysis. Lactose is an interesting story. Lactose comes from mother's milk, and in placental mammals, lactose is synthesized only in the mammary gland, and then only during late pregnancy and lactation. The synthesis is done by lactose synthase, a dimeric complex of galactosyl transferase and alpha-lactalbumin. Lactose breakdown in the intestines by lactase provides newborns with essential galactose. Some humans, actually about 2/3 of the humans on the planet, end up to be lactose intolerant due to a particularly low level of the lactase enzyme. Lactic acid fermentation by certain bacteria is the basis for the production of yogurt. This reaction shows you how lactose can be broken down to galactose and glucose by the lactase enzyme. This enzyme is present in newborns, but eventually most humans stop making lactase, and only people that descend from Northern Europeans, for the most part, retain the ability to break down lactose in this way. This slide shows you that the predominant persistence of lactase is from individuals from Sweden and Denmark and the United Kingdom and so on. Individuals from Greece and Japan and Native Americans have very low levels of lactase persistence. The last section in your text in Chapter 18 discusses how cells respond to hypoxic stress. Glycolysis is an anaerobic pathway. It does not involve consumption of oxygen. The TCA cycle is referred to as aerobic because it couples to oxidative phosphorylation. When oxygen is abundant, cells use both of these pathways in aerobic metabolism. When oxygen is limiting, though, cells adapt to carry out more glycolysis. Hypoxia, a shortage of oxygen, causes changes in gene expression that increase levels of glycolytic enzymes. A trigger for this is a DNA binding protein called HIF, the hypoxia-inducible factor. HIF is regulated at high oxygen levels by hydroxylase factor inhibiting HIF, also known as FIH1. Now, HIF consists of two subunits, a ubiquitous HIF1 beta subunit and a hypoxia-responsive HIF1 alpha subunit. In response to hypoxia, inactivation of certain prolyl hydroxylases allows stabilization of HIF1 alpha, also dimerization with HIF1 beta, and binding of the dimer to the hypoxia-responsive element, the HRE, of HIF target genes. Activation of transcription of these genes follows, and VHL, by the way, in the next figure is the von Hippel-Lindau subunit of the ubiquitin E3 ligase that targets proteins for proteasome degradation. This slide shows the essential features of the response to hypoxic stress. We won't spend very much time talking about it in this chapter. We may come back to certain elements of this in later discussions.